phenomenon is being observed? What phenomenon is being observed? The heterozygous individuals are indistinguishable phenotypically from the homozygous dominant individuals. What does indistinguishable mean? What does it mean? What kind of phenomenon is this? It's kind of a trick question. It's very simple. If you think back to chapter 5, right? That's what we call complete dominance, right? Think about this. What happens in a monohybrid cross? Right? Let's say you're looking at black versus white guinea pigs, for example. And let's say that fur color is coded by gene A. In the parental generation, we'll take two true breeding individuals, meaning we'll take a black individual and cross it with a white individual. What will the F1 generation look like? All heterozygous, meaning they'll be black. And then you will, to obtain the F2 generation, you'll self-fertilize the F1, you'll self-fertilize the F1, so black cross with black heterozygous, and the F2, what do you get? A 1 to 2 to 1, homozygous dominant, heterozygous to homozygous recessive, right? These three individuals are all black. 3 individuals are black, 1 white. Why? Because the dominant A allele is completely dominant over the recessive A allele. So that's why the heterozygous individual also looks black. You can't tell them apart. You cannot tell the heterozygous individual apart from the homozygous dominant. That was what we called complete dominance. That was complete dominance. But let me give you another example. Let's say you're looking at petal color in snapdragons. You're looking at petal color in snapdragons. And let's say for the parental generation, you took let's say that the dominant allele A codes for red, and the recessive A allele is for white. This is a red flower, and this is a white snapdragon. Let's say that the F1 individual looks pink, heterozygous. So what's going to happen in the F2 generation if we self-fertilize the F1 generation? What's going to happen in the F2 generation? What if you get pink in the F1? What is pink? It's somewhat intermediate between red and white. So the heterozygous individual is having a phenotype that's intermediate between the red and the white. So that means that the red is not completely dominant over the white. Simply having one dominant allele doesn't get you the complete dominant phenotype. This is what we call incomplete dominance. And if we self-fertilize the F1 generation, what are you going to get? You're going to get a 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive, but the phenotypic ratio is going to be 1 red to 2 pink to 1 white, instead of 3 red to 1 white, as we would expect. We would not have expected this pink individual. We would have expected a red individual, because from what we've already seen from chapter 5, is that if you have just one copy of the dominant allele, you automatically get the complete dominant phenotype. And obviously this is not what we're seeing. So that is why we will conclude that this is incomplete dominance. Chapter 14, incomplete dominance. The next question was, in the RBE2F pathway, what is the transcription factor? What is the answer? E2F. Very good. E2F. So E2F is basically going to be the transcription factor that's going to activate the genes responsible for DNA synthesis. But what keeps it sequestered, meaning blocked, is the RB. So what causes RB to eventually detach from the E2F? Anybody? what eventually causes the RB to detach, to finally let go? The phosphorylation by the CDK cyclin complex, right? Very good, excellent. Protein kinases, they phosphorylate their target proteins, whereas phosphatases dephosphorylate, meaning they remove a phosphate group. Protein kinases usually have an activating effect on proteins, whereas protein phosphatases usually have a deactivating effect. Adding a phosphate group to a protein usually causes a conformational change in the protein. What do we mean by a conformational change? A change in the shape of the protein, right? A change in the shape of the protein. And a change in the shape of the protein will likely affect what? Its function. Its function. So G proteins are very important. If you take cell physiology, you saw that Dr. Wilk talked a lot about G proteins. The reason G proteins are so important is because, well, one is simple. The sample says 90% of a lot of the medicines we use target G proteins or some related aspect of G proteins. So what do G proteins do? What are they? Let's talk about that.